Hello, my name is Chris Wetzel. I'm with the company MMI, and I would like to introduce our system. We have, for more than 25 years now, we have successfully developed and sold systems for cell isolation. And for cell isolation, that means if you have cells in tissue, you can use something that is called laser microdissection. And the microdissection you will see here, you have the laser in the back, and the laser will come through the microscope and come up here. And then you have a sample here, and in that sample you can then cut the cells of interest, and these cells of interest will be in these tubes, and then they can be analyzed for gene sequencing. Now this is well for cells in tissue. If you have cells in liquid suspension, then the things are not quite as easy, because then you cannot use a laser anymore. Or it would be like Star Wars underwater, but that does not work. So instead we have developed this capillary add-on here. So you can fold over a capillary onto the system. This capillary will then be able to pick up cells from a slide in the center, and here you actually see the laser microdissection part, and you can select the cells of interest in the laser microdissection. Like you can select the part here, and then you go to the laser, which I'm not going to do now, and you cut out exactly the cell that you want. You can easily move over to the selector part that I mentioned. You go over to another slide, and there you might see the cells of interest. Then you fold over the capillary, but you have to remove the laser microdissection part first, which I'm not going to do now. But then you can pick up the cells of interest, and they will be in the capillary, and they will be transferred into some other wells and for further gene sequencing. These are the two major parts and systems that we offer to isolate cells for your convenience. And we have a third system which won the Nobel Prize this year. No, not the system, but the person who invented it. And that's a cell manipulator and optical tweezer. The nice thing is, as you see, all these systems can be combined into one system, so that will be an optimal system combination for isolating cells for gene sequencing.